Hello, I'm going to present an imputation or denoising approach for single-cell Hi-C data with variational graph autoencoder. A high-throughput chromatin conformation capture assay Hi-C detects genome-wide chromatin interactions. The processed output looks like a squared matrix where rows and columns correspond to genomic bins and each entry of the matrix represents observed interaction frequency between a pair of genomic bins or a bin pair. Many interesting 3D structures like loops and topologically associating domains or TADs have been detected from analyzing Hi-C contact map. These structures play key roles in gene regulation. Therefore, it is interesting to detect these structures from Hi-C contact map, which requires robust measurement of interaction frequencies of a contact map. Single-cell Hi-C data has one more step for isolation of cells, which enables detecting genome-wide interactions at single-cell resolution. Multiple single-cell Hi-C technologies have been developed in recent years that output many single-cell contact maps per experiment. The major difference between single-cell and bulk contact maps is a sparsity, meaning much less interactions are observed in a single-cell contact map compared to the bulk map. The sparsity of single-cell contact maps is decreasing by improving single-cell Hi-C technologies. However, they are still much sparser than bulk contact maps to identify 3D structures from them. Imputation approaches share information across datasets to denoise single-cell Hi-C maps. For example, they use information from neighbor cells or neighbor breed pairs to denoise single-cell measurements. Different imputation approaches have different assumptions, and depending on those assumptions, the imputed contact maps can be very different from each other. Probabilistic modeling of single-cell data is interesting because it allows modeling uncertainties of single-cell measurements to learn denoise lower dimensional space underlying data and decoding denoise measurements from that space. Many deep probabilistic models commonly based on variational autoencoder have been proposed for learning the latent representation and imputation of different single-cell data modalities like single-cell RNA-seq and single-cell ATAC-seq. They encode a high-dimensional vector representation of a cell into lower-dimensional latent vector and then reconstruct the noise high-dimensional vector representation of a cell from that. However, such models encode the vector representation of a cell and they are not directly applicable to single-cell Hi-C data where we have a matrix representation per cell. Single-cell VI 3D is a current generalization of VAE-based framework to single-cell Hi-C data. It starts with a band transformation where they divide each contact map into several bands where each band is a diagonal of a matrix. Then they concatenate each band across cells separately, resulting in a cell-by-feature matrix per band, and train a model for each band. However, this approach is going to ignore spatial dependencies between Hi-C features by separating bands from each other. On the other hand, variational graph autoencoder is an extension of VAE concept to graph data. It also has encoder and decoder, and the encoder takes the graph's adjacency matrix and encodes it into the latent space, which is usually implemented with graph neural network, and the decoder is going to reconstruct the graph's adjacency matrix from that space, which is usually implemented with similarity function like inner product. ContactVI utilizes VGA architecture to denoise single-cell Hi-C data. It assumes that observed single-cell measurements are drawn from zero-inflated negative binomial distribution parameterized with expected interaction frequency, dropout probability, and a dispersion. For expected interaction frequency, it applies a softmax layer per pool, which is a sequence of bands, to normalize expected contacts per pool. It has another module to predict the dropout probability given sequencing depth and a genomic distance as inputs. It also learns a common dispersion parameter. For the evaluation of imputation approaches, we use single-cell imaging experiment, which is a low-throughput orthogonal assay to single-cell Hi-C, which directly detects 3D coordinates of the genomic bins within a specific genomic region. This Distance measurements are much less noisy than interaction counts from single-cell high-C data. We assume noisy single-cell imaging data as a ground truth and construct ground truth map from that and also simulate a sparse contact map with downsampling from that. And then we calculate the similarity of imputed downsampled and ground truth contact maps. Here we can see that ContactVI performs better or comparably to other imputation approaches in reconstructing ground truth single-cell contact maps. There is another technology, single-cell combinatorial indexed high-C, used for profiling human cell lines at single-cell level, for which we also have enriched bulk high-C maps. We calculate the similarity of imputed single-cell contact maps and enriched bulk high-C maps. Higashi neighbor is going to average neighbor cells for imputation, so we expect that to has the highest similarity with the bulk map. However, ContactVI is going to outperform other imputation approaches in reconstructing ground truth bulk maps. As a conclusion, single-cell high-C imputation methods improve the similarity of single-cell profiles and ground truth profiles. Deep probabilistic models enable modeling and denoising stochastic single-cell measurements, and ContactVI is going to use a deep probabilistic model for a graph structure to impute single-cell high-C data, and it outperforms or performs comparably to other imputation approaches in reconstructing ground truth single-cell maps. Thank you very much for your attention.